Hello and welcome to our C32 Defense Against Disease section. This section starts here with just a simple definition of a pathogen. So what is a pathogen? A pathogen is a disease-causing organism. There are many different categories of pathogens. There are viruses, bacteria, fungi, and protists. So these are all different organisms that can cause a pathogen, or cause a disease, excuse me. And so protists are single-celled eukaryotes, fungi, right, are eukaryotes as well, then bacteria, and then viruses are non-living. Archaeans do not cause disease as far as we know. There are two nature of sciences, or NOSs, that you're supposed to know for this, is that careful observation is super important for solving diseases. Specifically, it was used twice in the past, once with childbed fever in Vienna. And for this one, they realized the importance of hand washing. So basically, they weren't washing hands in hospitals, and they were killing pregnant mothers during childbirth. And so this was Semmelweis was the one who figured this one out, and he made observations and realized that if you wash your hands, more lived. The other one was our cholera in London. This is Jon Snow, the hero of the North. No, different Jon Snow. And so what we've got going on here, cholera in London, and this one was treating the water. So they realized that there were diseases that were being caused by the water, and so water treatment lowered the number of diseases. Standard 2 is, well, how do we stop diseases? This is our skin and our mucous membranes. And so these two act as a barrier to defense. They act as a barrier, so they stop the pathogens from entering completely. So very effective. And then for our skin secretes a substance, and that substance lowers the pH. So the skin secretes a substance that lowers the pH, and our mucous membranes secrete mucus to trap pathogens. They trap it, and then they can also have cilia to expel it. So cilia are this is a cell. They're the microscopic little hairs on the outside of it, and those wiggle back and forth to help move it out. Standard 3. What happens if you get a cut in that skin? And so if you get a cut in the skin, there's a cut, and that cut happens, and then that is going to cause platelets to release clotting factors. These clotting factors start a cascade. The clotting factors trigger conversion of prothrombin to thrombin. And thrombin is an enzyme. And that thrombin then takes fibrinogen and converts it to fibrin. So fibrinogen is soluble, so it's dissolved in the blood, and fibrin is insoluble. And so fibrin then forms a sticky mesh. And that sticky mesh traps blood cells and platelets, creating a blood clot. So that is standard 3. They said in the standard, blood cells, these are known as erythrocytes. So blood cells and erythrocytes are the same thing. Erythrocytes. Great. All right, at standard 3. Standard 4. So we have two different types of immune systems. We have innate and adaptive. Innate is all the time, and it's non-specific. Adaptive, on the other hand, is specific. Specific. And so non-specific and specific, and then so this one is responding to a specific pathogen. And this one you can have memory, and so you can react faster in the future. So this one is very different. So this one all the time it's non-specific, and it's like, go attack it, whereas this one is like, oh, I recognize that, it's this, we kill it with this specific antibody. I don't know why I made the table so big. That's pretty much the extent of the differences, because we're going to talk about them more, really. So for the innate immune system, the only thing is standard 5, and that is our phagocytes or macrophages. These are amoeba-like white blood cells that look like this, and they're filled with lysosomes. And so what they do is they ingest anything that's not self. Engulf anything not self. So they engulf anything not self and destroy it. And they destroy it using the lysosomes, which are filled with lysozymes. If you're in HL, we learned about this in our organelle unit. So the lysozymes are trapped inside the lysosomes to prevent them from digesting the inside of the cell and to keep it safe. And so they digest it. And then one of the things they do, and this is like the end of the innate immune response, is they do something called antigen presentation. So antigens are the markers on the outside of cells, the glycoproteins that they can recognize. And so you have a cell like this, it's got like a glycoprotein marker on it, or it could have a marker that looks like this. And so it recognizes that and it goes, oh, that's not self, and we will attack it. Okay, so that's our antigens. And so this is really leading us into like standard 7. So antigens are glycoproteins on the outside of the cell. And so after they digest it, what the blood cell will do is it'll take those and it'll put it on the outside of the cell, and it'll go start the adaptive or the specific immune response. And so what we have happening now is we have standard 1, 5, 7, 6. So it's all about lymphocytes now. So the first part is about our phagocytes or our macrophages. That's the innate immune response. Now we're getting into the adaptive immune response. So our adaptive immune response is all about lymphocytes. And the end result is that we're going to make specific antibodies. So we are going to go to step 8. So step 8 don't forget, right, for standard 7, red blood cells have antigens as well. And so if you have A-type blood and you get B-type blood from a donor, you'll attack all those B blood cells. If you have B-type blood and you get given A, you'll attack the A blood cells. O can only get O blood because of the markers. So just something to remember. Okay, that's standard 7. Now we're done with that. Now we can go on to 8. So the macrophage activates helper T cell. A specific helper T cell, excuse me. from there, and it does this through the antigen presentation. So it sees the antigen. Then what it does is that helper T cell activates specific B cells, or B lymphocyte. They're the same thing. So T cells, B cells are lymphocytes. So when we say lymphocytes, they can be T or B. Okay. So T cells come first. The helper T cell activates the specific B cell. And then that plus seeing the antigen, it has to see the antigen as well, will turn the B cell on. So it requires both helper T cell and the antigen. So those two together will activate it. Now, activated B cells can become one of two things. They can become either a memory cell, which is standard 10, or they can become plasma cells. And so if they do this, they undergo proliferation, which is the fancy way of saying lots of mitosis, to make many, many copies. So they make lots of B cells, B plasma cells. So B plasma cells are activated, and then those will make antibodies. Antibodies are Y-shaped proteins, also called immunoglobulins. Okay. And so these antibodies then attack pathogens. And so they're all the same antibody though, the same antibody over and over again. And that same antibody is specific to the specific antigen, so it recognizes just those markers. Okay. The memory cells give the long-term immunity. So these memory cells live in the blood and they float around and provide long-term immunity. So they're very long-lasting. Okay. So that is the adaptive immune response. All right, then we just have some kind of other diseases that they specifically want you to know about, starting with HIV. So HIV has two standards. The first one is the transmission, and then 12 is the consequences. So the transmission of HIV is through bodily fluid, or body fluid. Right? So what does that mean for us? It means that it can be transmitted through any type of specifically body fluid. So not spit or saliva, but it can be transmitted through any of the other main ones. And so those would be things like blood, semen, rectal fluid, vaginal fluid, or breast milk. And so what happens is it's in the blood, and then it happens. The two main ways are unprotected sexual contact or some sort of needle usage. And so that is contaminated needle. So those are the two main ways you can transmit it from those fluids. From there, what actually happens? Well, when the virus enters, HIV specifically targets helper T cells. Okay? And so it's infecting helper T cells, and targets and infects helper T cells. Now what that means for, this is standard 12, what that means for us, right, is you have these T cells, so it's macrophage, T cell, B cell, antibodies. And this one, you're attacking these cells. So this part of the process can no longer happen because HIV is infecting right here. And so you're stopping this cascade from happening. And it leads to AIDS, or autoimmune deficiency syndrome. And so because you don't have enough helper T cells to activate your innate or your adaptive immune response, then you have that happen. And what that means is you can die from other infections. So once you get AIDS, your immune system is typically really weak, and other infections that a otherwise healthy person would normally fight off, you can be very susceptible to. And so this can lead to death from things like pneumonia or tuberculosis or salmonella, something that would not normally be a huge concern, but can be much more concerning if you have HIV. So that is standard 12. Right, then we have our antibiotics. And so this is standards 13 and 14. So antibiotics are for bacteria only. Let's start with that. Bacteria only. The only thing they affect is bacteria. And they target bacterial cell wall synthesis or bacterial metabolism. These are the main things they target. You can target other things, but these are the main ones. And so the cell walls of bacteria, remember, are made out of peptidoglycan. And so that is very different from plants, which are made out of cellulose. And so you're stopping these from happening. And then you kill the bacteria like that. Now, viruses, no metabolism, no cell wall. So therefore, antibiotics don't work on them. They have no metabolism, they have no cell wall, so you can't target that with antibiotics. Eukaryotes, right, have a different type of cell wall, and they have eukaryotic metabolism. Therefore, it doesn't affect them either. And so this is why eukaryotes are safe from antibiotics like us. So when we take antibiotics, it doesn't hurt our own cells. It does hurt the good bacteria in us, but that's like different. It's not a big deal. Well, okay, let me rephrase. It is a big deal, but it's not. There's nothing we can do about it, at least at the current moment. All right, then we have our evolution of antibiotic resistance. And so this is a good example of natural selection. It's a pretty common IB question, right? How does antibiotic resistance happen? Well, you have a population, and in that population, there's variation. Some of them have a mutation that allows them to survive antibiotics. And so what that means is, for some reason or another, their pathway is different than what you would expect. And so there's population variation, and some of them have this mutation that allows them to survive. And so if you treat with antibiotics, all die except those with the mutation. And those survive, and then they reproduce. And now you've got a large amount that are resistant. And so the more you use antibiotics, the more likely it is that you're going to use it on a population that has a mutation that can survive. And so you increase the likelihood of creating a large amount of resistance every time you give out antibiotics. The other issue, right, is if you don't take a high enough dose and you don't take it for long enough, this also can lead to multi-resistant bacteria because you didn't kill all of them, so they were able to recolonize, and now you have a bigger issue than you did before. So that's why doctors want you to take it for the whole time, even when you feel better, and it's why we try to not prescribe antibiotics for things that won't work, because you run the risk of increasing it. Okay, next, we have our zoonoses. Zoonoses are, well, they're not cool, they're terrible for us, and they're getting worse because the barrier between humans and animals is getting less and less as we move into their habitats. But it's infectious diseases from animals to humans. And there are a lot of these. There are a lot of them. And so it depends on which one we're talking about. So tuberculosis can jump from like cows in their milk. It can be found in cow's milk, and that can affect humans. Rabies is from a bite. Japanese encephalitis is like West Nile, it's a mosquito. So this one is a vector-borne, and so the vector is the mosquito. So the mosquito doesn't actually get sick, it's the waterfowl. And this one is other small, it's mammals. And so what happens in that one is, right, it's the mosquito bites a bird and then it bites a human and it transfers it that way. That's the same as West Nile. Other ones, right, is from like food, and so Ebola is from bats, and so that's another one. Same with COVID is also from bats, and that one is like eating it as well. And so you can eating it or just coming in contact too, right? So those are both possibilities. So those, what they do is you take this disease that was an animal and it jumps to humans. Same thing's happening with avian flu right now. And so these are really bad because they jump and then they can cause pandemics, right? So we know this is what happens. It's cause pandemics, and then unfortunately, if those we're not careful, those become endemic. And so that's what we see with COVID. All right, 16, vaccines. So one of the things we can do, right, is a vaccine. And so a vaccine typically nowadays is one of two things. It either has antigens or it has nucleic acids to create antigens. These are our newer ones. And so either of these, what they're doing is they're activating the adaptive immune response. And so what you're doing is you're turning it on, and now you're creating memory cells. You're creating memory cells because you've activated the adaptive immune response. So you go macrophage, helper T cell, B cell split, and some of them become memory cells. And so that helps give you immunity without the disease. It's very effective, and it helps lead to standard 17, which is herd immunity. And so herd immunity is when, if you have enough of a population is immune, it slows disease transmission. There are two ways to get to herd immunity, and one of them is better than the other. One of the ways to help get to herd immunity is our vaccines. The other one is to get the disease, right? And so obviously there is a lot of dangers in just letting a disease spread. And so there's a lot of long-term side effects from getting disease. And so we attempt to vaccinate whenever possible to help slow it down. All right, now this leads us to like making vaccines, and so then you got to be careful, right? There was a lot of misinformation about the vaccines when they were being released, and so it's about deciding, like you got to look at the research and know that the media looks at the research while it's happening and before it's been published. And so typically what we like to do is wait for the research has been published and then we make the decision. That's the nature of science standard for this one. All right, last we have our COVID-19 pandemic data. And so you're supposed to be able to look at the data and compare. And so there's both percent difference and percent change, or percentage change. So both the formulas you multiply by 100, but for a percent difference, and so for a percent change, or sorry, percent difference, what we're going to see is that it's final minus initial over the average times 100. So that gives us a percent difference. The average is final plus initial divided by 2. And so that's that one. The other one is final minus initial over initial times 100. This one will always be smaller, right, because you've averaged them first. And so this one is how much has it increased from the initial. This is how much has it changed. How different is it percentage-wise? And so it is, they're looking at different things. Okay. Percent difference is the difference between the two values, the average of the two values, whereas percent change is how much it changed from the initial. The percent change one is more common. I don't see a lot of percent differences, but maybe they'll start adding it to the IB test, because I don't really remember it being on the old curriculum. So it's possible they'll add it. These could definitely show up on the database questions though, and so you definitely want to make sure you know both of these formulas. And that is C 3.2.